And the last thing I want to show you today is two analysis modules. One is the mini detection, event detection, and the other one is action potential analysis. Here we got a series of miniature postsynaptic events. And what I'm going to do is not analyze it, but do a right click. Or we could also do a context click up here. That's the same thing. The menu content changes depending on the level, the type of node that we have in the data navigator, the type of node that's selected. We have the paradigm P2 in this case. We have the 8th routine, R8. And within that routine, we have signal 1. And event detection is something that takes place on the signal level. We do that with individual signals. You would not do event detection at the same time in a current signal and a voltage signal that you may have in the same series. So let's start the event detection here. In the template field, we can create a template defined by rise time and decay time. That is shown here then. We can set the threshold that's expressed in times standard deviation. And if we had elicited events, we could, within this data set that has 40 sweeps of 20 seconds duration each, and if it was elicited, we would know anything before the pulse that elicits our responses, our events in this case, is invalid. So you would start at, say, 5 seconds or something like that, and that way also avoid the stimulation artifact. But in this case, this is all spontaneous postsynaptic events, so I will just start with the analysis, and it's calculating. It's running the deconvolution algorithm that detects the events, and here we have a double event, and this deconvolution algorithm is powerful enough to detect the second one in this double event. So we can go through and look at all events individually, adapt our template accordingly if we want to. That may be easier if we average, but before we average, actually, let me remove a small one. Let's assume this one wasn't valid, so I want to remove that from further analysis, and the same with this one. And then what I'm going to do is I'm going to average, and here we could see, okay, yeah, our decay could be a little faster to match the actual averaged waveform here. We can save the results, and what that does is it creates two things. It creates a table with the results, with the numerical results. For each of the 750 events, it has an entry in this table that can be further processed either within Igor or in a third-party application of your choice. And the second thing is it creates a layout that you can use for your workgroup presentation, or you can modify it similar to what I showed earlier and use for the figure in your publication. Same thing here. Show this, make the trace black in this case, and you got your cumulative mini amplitude histogram. The last thing I want to show you today is action potential analysis, and we want to start in this case with spontaneous action potentials. Same thing, I'm going down to the signal level. Signal 2, the voltage signal, contains the action potentials recorded in current clamp mode. And here we'll look at the action potential analysis. We set the threshold, 0 millivolts is appropriate because every action potential exceeds 0 millivolts, and everything that exceeds that threshold looks like it's an action potential. We could also define the event length. And we can calculate the action potential duration at 50, 80, 90, or 100% repolarization. Let's start the analysis again. Here we have 2,000-ish events detected. We can look through them. They look pretty similar, which isn't surprising. They are spontaneous, so nothing changes from one to the next. And here we have the phase plane plot, the dV over dt versus the voltage. And that is a pretty sensitive way of plotting the action potential waveform that's pretty sensitive to changes in the waveform. And again, things remain pretty stable throughout here because, as I said, not that much changes. Same thing here, if we average the results, we get the average phase plane plot, the average action potential waveform. And if we save the results, we get tabular results and a layout that we can use to keep track. And if we do the same thing with not the spontaneous action potentials, but elicited action potentials, these action potentials were elicited, let me analyze that real quick, were elicited by current steps, by depolarizing current steps. I'll disable the persistence display here, which shows only individual sweeps. At the 80 picoamp level, we start getting action potentials, and then at higher currents, the frequency increases, and then we'll just close that, and I'll do a right click and go action potential analysis, and I'm running the same action potential analysis here, except now I'm running it on elicited action potentials, which means I expect changes in the phase plane plot between individual action potentials, particularly the first one in a sweep. Like this one, I expect to be very different from the consecutive ones, and then the consecutive ones are more similar. And averaging in this case obviously doesn't make any sense. This is what I wanted to show today. There's a lot that we have not talked about yet. For example, paradigms, the planned paradigms that I talked about in the data structure. We haven't talked in too much detail about metadata and how to work with them. We have not looked at the current clamp portion of the amplifiers, and this is somewhat different between the IPA and the D-patch systems, and the I/O, which lets you control periphery either through analog or digital outputs, and you can do that either, this is in essence setting the holding levels, but you can also define waveforms as part of a routine waveform. But that will be shown in a separate tutorial.
Follow along the next episode in the series, or jump to the playlist for other informative videos. Subscribe now to keep up with the latest installment.